Thank you for that nice introduction, and thank you for the organizers to let me speak here today. Let me figure out this. Oh. So I'm going to talk about metagenomic NGS and the massive data sets and why K-mer algorithms are very popular in that field, and why we moved to an MNGS K-mer algorithm. And then talk about like how we benchmarked and actually validated it. Last week, we actually completed the validation of this pipeline. So I'll show you how we did that and results and discussion. This is just like a quick overview of like metagenomics NGS. So you start with a sample. You don't have to culture it. That's it's a culture independent method. You extract the nucleic acids, you sequence it, you get a lot of sequences, and then you start to figure out what the taxonomic source is of each sequence. And you see like the different colors here mean like, the same taxonomic source. So we already have a pipeline or, the previous pipeline. So why are we moving to a new, algorithm if we already have one? Especially clients are like, why are you changing the algorithm? So there are multiple reasons for that. But one of the biggest ones is that the growing volume of data has driven the development of smarter algorithms. So if you look at like BLAST, for instance, when that came out, we talked about kilobytes of data. These days, we talk about terabytes of data. And you see that as databases grew in size, as like the throughput of like, Illumina machines and all that grew, you saw that the bioinformatic pipelines dealing with that data also changed. So first it was like local alignment, read mapping, that's like BWA, Bowtie 2, I think you're familiar with those. Then around 2014, K-mer tools came out, like FairFinder, Kraken, STAT, which is like from the NCBI. Deep Microbes is K-mer and machine learning combined, but you clearly see like how the evolution of tools correlates with the growing volume of the data. And so we had to adapt as well. So this is just looking at Merck. So back in 2010, we had our first AAT algorithm, but at the time we dealt with 4x4 data. We had like 1 million reads a run on average. And so we used a local aligner BLAST, which was perfect for that data. Then in 2017, we actually had our first validated AAT pipeline, but we used MySeq data, and that was already an increase in the throughput. So BLAST didn't really work. So then we switched to a read mapper. Now we are using NextSeq 2000. We're talking about like 400 million reads. We're also thinking about acquiring a NovaSeq. So then we talk about billions of reads. So we just have to update our algorithms from time to time rather than, acquiring more RAM, more CPU. Like you have to change your algorithm from time to time as well. So that's what we're using right now, like a metagenomic classifier. So our current AAT pipeline, even though I just said the K-mer one is validated, current, I mean the one that we're using right now, the read mapping based approach, is at max capacity, as you see on that gauge, because our database is like over 1 terabyte in size. Building that database takes a lot of time. We also have a lot of like redundant results. So like one read that hits multiple accessions or multiple taxa. So for study management, that's a lot of data to look through. As I said, it's computationally intensive, and it's only limited to small genomes. So like if we want to look at bacteria or fungi, the databases are even smaller. And we could technically run it, but then we have to run it on an even more expensive cloud instance. So various reasons to switch to a K-mer algorithm because of that. So the revised pipeline that uses K-mer under the hood, as you see, the capacity is now in the green. We use a smaller database because we only store diagnostic sequences for each of these viruses. And so that really reduces the database, but also the time to build these databases, to screen against these databases. We don't have duplicate results, so it's just like one line per taxon. It's more binary decision-making for study management, and we're able to also analyze bacteria and fungal data sets. So I don't have an overview of like, because the legal department didn't want me to add it, like how it works, like the pipeline, but we also, we can take out like cell line. We of course do like retrimming at the beginning. So all those bells and whistles are there. And then we create like a C of A, or like a certificate of analysis, like a draft that's also automatically generated. So I think most people know, but just in case, like, what is a k-mer? I know it's like, it's an old term, but it's basically just like a sequence of length k. It's very often length 31 because that's like very optimal for like genome assembly. It's an odd number also. And it can also be created for like proteins, of course, not just nucleic acids. So as I said, it's being used in many tools. If you have, for instance, this sequence and you want to like, create a trimer, then you start at the beginning and you move that one position up until you reach the end. So you always move with the stride of one to get a good k-mer coverage of a certain sequence. Yeah. So why are these k-mer tools so fast compared to read mapping or like a local alignment, right? So let's say you have this sequence in blue, and then you have that sequence in purple, like in your database. And you see in red, there are a couple mismatches, right? So with k-mer matching, you're just asking, are we 100% identical or not? And if not, then you move on to the next one. So it's just like a binary decision for all of these. And computers love that type of stuff. It's just like yes or no, yes or no. Versus a read mapper, of course, it's going to look like, okay, are we within certain parameters? Can I create a mismatch maybe? Like, can I extend the gap? That takes time, especially for millions or even billions of data sets. And that's just one part of the workflow, because you also create massive BEM files. You need to go sift through that as well. So like the downstream analysis, in my opinion, are worse than the actual read mapping. You heard it yesterday, Mark was mentioning it as well. So the lowest common ancestor is something that we use in our pipeline as well, because not every k-mer is going to uniquely map to a particular taxon. So again, you have this sequence. And what if it hits both Zika and hepatitis C? We're not going to then count it for both of these. We're going to look at the lowest common ancestor, which is like, what do they have? Where are they basically in common? And in this case, it's the I'm not going to pronounce that one, but that family. And so then, yeah, if you only would have this k-mer, the only thing we can tell is like this sample contains that family. But luckily, we have way more k-mers, and so you will get species and strain results. So as I said, we just finished the validation of this pipeline last week. So how did we do that? So we took data, which is being discussed by Brad already. Rebecca has a poster on it. Alice was talking about it yesterday. We took this data from Rebecca, that's published in Biologicals. And what they did is they used 4 cell lines that are commonly used in biomanufacturing, HeLa, CHO, Vero, and SF9. And we spiked in 3 representative viruses, RNA viruses. I have to say, we're now also going to validate our cell-free assay and like transcriptomics. There we will use the WH7 panel. But for the bioinformatics only assay code, that's what I'm talking about right now, we did it on these 3. So we did, so actually Rebecca's group used 4 spike levels from 1E6 to 1E3. But when we started analyzing this data, we were able to detect everything in 1E6, everything 1E5. So we were like 1E5 is for validation, our highest concentration, and then went down. We did everything in triplicate as well. And then the LOD is the lowest dilution in which the virus was seen in at least 2 out of 3 replicates. But now for the new validation that I was just mentioning for the other assays, it will be a 3 out of 3. And so we use our new, we used the new k-mer algorithm, and we also do like data qualification, which is, we'll look at all the positive hits in the context of the sample, and then, see if it was actually like a true hit or not. So, for looking at the specificity, you saw like the unspiked samples, we didn't detect any of the spikes. So you use 0 out of 3 for all of them. And for the highest concentration, which was 1E5 spikes, we were able to detect these viruses in all 3 replicates in all 4 cell lines. And so the limit of detection, you can look at it like differently for like each virus. So for RSV, we were actually able to detect it to like 1E3, and then feline leukemia and RIO1, 1E4. And then we say basically, so for all of the cell lines combined, basically we then say that 1E4 is our limit of detection, because if you look at like all of these together. And so here's just a table that shows, we were able to detect everything 1E5, 1E4. We had a couple where we, didn't detect Rio in one of the replicates. On the species level, I get there. We did detect it, but at the family level, but I get there how we find that out. But on the species level, 2 out of 3, and then you see 1E3 gets a little bit spotty except for RSV. But this is all like on the species level. But let me show you a little bit. So here, for instance, before a couple deep dives. So here you see at the top are 3 replicates for the Kamer pipeline, and at the bottom you'll see the read mapping algorithm, and then you can see how they compare. So if you look at RSV, you see that we detected less reads compared to like read mapping, but that is because as I was saying, these Kamers, they're specific to a certain taxon. If you look at it that way, of course, not every Kamer is going to be specific to a certain taxon, so you always find less. You see the same in machine learning. I don't know if people saw my talk last summer. Of course, the Kamers that are specific to a certain taxon, they are weighted more than like conserved sequences that are found throughout the viral kingdom. So yeah, you always find a little bit less reads, but that's fine for a qualitative assay. Then FELV, you saw that we detected more, and I get to that why that is. And then Rio was like more or less the same. And so here you basically see we used a human genome filter. Then you see FELV comes down to like where the read mapping was as well. And we looked at like what was happening. Of course, when you use Kamers, they're shorter sequences. And so they are, of course, less specific than a full read. And we saw that the cellular genes in feline leukemia were actually, cell line reads were actually hitting FELV. But when we take out the human genome beforehand, that doesn't happen. And that's why in SF9, you don't see that effect. It's a little bit more, FELV is similar between the two because SF9 is an insect cell line. And you see here, as I was saying earlier, Rio was missing, where it was picked up by the read mapping pipeline. But I get to that, like how to deal with that. Yeah, and this is like the same. So like human genome filtering, again, we missed a Rio species result. So that data qualification, as I was mentioning earlier, so that's part of the workflow as well. So what we do is we say each positive result that comes out of the pipeline, we need to take a closer look at that, right? Like at the context of the matrix. And by that, we mean each positive result. So not just species, but also family or order results, because those are also, they could be something. Because with Kamers, at least, I think Robert has a different approach to that, but at least in our pipeline, we don't know where the Kamers belonged in the genome. So we don't know the position. But that is necessary information because you want to know, for instance, like are reads distributed across the genome, or do they pile up on one region? Or for instance, if you have the HeLa cell line, you're going to find HPV 18, but only the E6, E7, and E1 genes. If you find more than that, that might be an infectious HPV 18, but Kamer will not tell you that. So during qualification, we're actually looking at these reads. And then you can, for instance, see, oh, in this case, you see in blue at the top, like where the coverage is, it's at the beginning only of the genome. So now you know, like, okay, the K-mers that hit HPV 18 were actually from the integrated HPV 18, not an infectious HPV 18. So this is just to show how data qualification works. So you see here like F being family, G genus, and S the species, and S1 like strain, for instance. So you'll see since now you look at like the whole reads, you can move, for instance, a family result up to species. So you're basically increasing your taxonomic resolution during that step, or that the species moves to, strain result, a species result moves to a strain result and all that. So, and this, again, this is like mandatory part of the workflow. So that's why we'll find these. And so again, I was showing you earlier here that Rio1, we didn't detect it on the species level. But then with the qualification, you see here, then actually we pick it up again. But that's a way to like deal with like, K-mers are slightly less specific than read mapping, but if you combine it with the data qualification step, you can actually overcome that. I also want to mention that, of course, we update our database every 2 months. So we actually increase our genetic diversity every 2 months. And the more genomes in our database, the better K-mer is becoming, of course. So if we would have done this 15 years ago, I think K-mer would not have worked. Like it really needs the genetic diversity. So speaking of the genomic reference database, I was actually talking to someone from ATCC, Jonathan Jacobs, and I know someone from his team is here in the audience. There are a couple of problems with reference databases. I know I also talked about it with PSU. One of them is metagenome assembled genomes, MAGs. You might have seen them. It's only 1.8% of our current database, but they do cause problems. We see that they're mislabeled, like there is a PCV1, but it's labeled as a PCV3, and that can really mess with your K-mer results. And I thought, like, they're only in GenBank. So, okay, if you just take them out, it's fine. We go with RefSeq. NCBI now says that they allow MAGs to be in RefSeq, which is quite horrible, but yeah. And as I was saying, there are models, but a model is only as good as like, the next model. And I feel like if you have a PCV1 genome, why would you also want to have a PCV1 genome in MAG form? Like it doesn't add anything. Also, if you look into like how MAGs are created, they sometimes pooled hundreds of samples from all, like Illumina, different library preps into one sample to get enough, coverage. And then people create like genome assemblies of that. So they're problematic. We for now just take them out, but that might be too aggressive. But like, yeah, it's a problem, especially for K-mer analysis. And then partial sequences as well. We have seen instances where we had, like unspiked samples, but we did find FELV at the beginning, and we're like, how is that possible? And then we saw that like in GenBank, it was a partial sequence. It was actually the human receptor for FELV labeled as FELV. So if you take out that partial sequence, then unspiked data looked as expected. That's just the tip of the iceberg. I'm not part of FT Group C, but I should. But yeah, so that's, let's talk about that more. But I do feel that K-mer analysis are more sensitive to, troubles with the database than read mapping. So but you just need to be aware of that and do your curation. So in conclusion, the metagenomic pipeline or the K-mer pipeline that we put in is really to future-proof our AAT operations. I also want to say, that the overall LOD is 1E4 per a million cells. And that, as I was showing, cell line genome filtering reduces false positive detections, like for instance with FELV. We made it an optional step. So we didn't validate using genome filtering because then it would have been, mandatory. We just keep it an optional step. And if for whatever reason genome, the cell line genome shouldn't be removed, then that's not a problem. But read mapping, that might be tough because usually 99.9% of your sample is cell line. If you take that out, then just the needle, that's perfect with read mapping. But if someone is like, I don't want you to take out cell line, then read mapping becomes an issue. And K-mer is like, yeah, that runs for 3 hours and then it's done. So as I was saying, the data qualification is increasing the taxonomic resolution of these data, and therefore increasing the relevancy as well. So that's it.
Thank you to the organizers for allowing me to have this opportunity to present this work on behalf of our team. So I'll briefly cover the viral metagenomic analysis that we're using to de-risk our live virus vaccines, how we've used bioinformatic analyses in the Hive tools, and our new tool called Virus Screen that's internal, and then a benchmarking study and comparison of workflows. So this has been covered extensively by this time in the conference, but really just want to highlight that we can use NGS to de-risk viral vaccines earlier on in R&D, and then as it steps through clinical development, there's an increased business risk that can then also be transferred into a commercial supply risk, right, and then affecting public health. So we apply a risk assessment prior to beginning a metagenomic analysis study, and that will help us determine the kinds of study samples that we have, the test articles, the controls, and whether or not it's a viromic or transcriptomic analysis. We determine the virus recovery using the CBER FDA virus panel, and then from Illumina short reads that are paired end, and then apply bioinformatic analysis. So again, this has been covered. We're using the 5-virus panel for determination of recovery. You'll also see I've used a rhinovirus and a porcine circovirus 2 in some of our data, and these are just used in silico to generate synthetic reads. So a brief overview of what we're calling the short read alignment workflow, or SRA. After sample preparation, extraction, and sequencing, we then process the reads for quality, and then the read sets from samples that have vaccine virus in them are stringently aligned to the vaccine virus reference sequence itself in order to basically subtract those out from the subsequent analysis. So the resulting reads are then aligned to a curated version of the RVDB that has been appended with the vaccine virus sequence as well in order to control for like cross-contamination or minor variation from the vaccine virus sequence. So then once we've done that, the best hits within taxa are reviewed across the study samples, and then the putative positives are counter-screened, and then anything that's bioinformatically confirmed will then need to be investigated. So we have used Hive to perform bioinformatic analysis for characterization tests in regulatory filings. So this high-performance integrated virtual environment has several instances of public domain. It's maintained by the experts at George Washington University, and it has well-known and published tools like the Hive Hexagon Aligner and the Heptagon Alignment Profiler. And it can also generate biocompute objects to communicate our pipelines. So this is a very manual process. We manually input read sets, and then we also have a command line curation of the viral reference database, input that as well, align it with Hexagon, profile it with Heptagon and Hive, and then those results are then processed taxonomically with very complex Excel worksheets in order to basically generate giant pivot tables that group the references by taxonomy, allowing us to determine the accessions within taxa that have the maximum coverage for species sequences and then the maximum total number of alignments. So in order to move forward with automation and streamlining of data analysis for GMP or release testing, we've developed an internal application called Virus Screen that is very user-friendly. It's within our company firewall, so we don't have to copy data around several times. We have the ability to customize the workflows in a non-GMP and GMP environment, and we can download a report, which is great. So we have multiple workflows available for paired end Illumina read sets. Today, I'll just talk about database curation, short read alignment, and de novo assembly alignment. So we curate the FDA clustered RVDB in order to reduce false positive hits. So basically, we have bulk virus harvests for the vaccines that contain lots of sequences from cells. So we are curating the database to mask undesired regions. This has been described in a couple of publications, but basically we split up those genomes into pseudo reads, apply classification for undesired reads such as like vectors and bacteria. And then we also use dust masker to mask low complexity regions. So ultimately what we end up with is converting all of those undesired nucleotides to ends, so we retain the same number of records. So this paper published earlier this year shows using simulated data sets how we are able to reduce false positives down to basically zero when we have curation. And so we are able to put this into the automated environment so we can just point and click, curate the database, and then we also have a summary that is generated, shown here as an example of what was masked with what tool. So we kind of keep track of that. So what are we automating exactly? So we're automating the steps of the alignments. And so we basically start out with a fast QC for all the read sets. In blue, if we have vaccine virus in the samples, then we perform that alignment first. Then we go through and perform bow tie alignments to the curated RVDB. And then abracadabra, custom Python scripts, several steps. We get really great tables that give us the best accessions hit for each species or genus and family, different taxa. And we're also able to get coverage maps and contigs and all kinds of goodies that we can use for follow-up. So I'll briefly describe this case study where we benchmarked virus screen GMP against Hive. So really what we're doing here is using research data sets spiked with either known virus amounts or known virus sequences for comparison, performing the short read alignments to the clustered RVDB version 20. So it's a little dated with the added curation. And then using hexagon alignments in Hive and bow tie 2 alignments in virus screen. So really we have data sets that represent in silico spike data where we have 22 million reads of background media read sets. And then we've gone in and spiked in silico generated reads that are 10 paired end reads per 5 viruses. So it's 20 virus sequences for 5 viruses. So that's one data set we're using. We're also using internally generated data of vaccine harvests and control harvest for a early phase SARS-CoV vaccine that we had a few years ago that was generated in Vero cells. So here we're spiking those with 1 × 10⁵ genome copies per mil of the FDA panel. And then lastly, we applied this to the AFTIG collaborative study HeLa cell that were spiked with 4 viruses at 3 different levels. So what we see is that when we look at either the maximum hits or the maximum coverage across either Hive or virus screen, that we really get very similar data across the different viruses that we're using here in silico. And then when we looked at this for actual spiked viruses into samples in our labs, we again have very similar coverage and number of hits. And then for the HeLa cell that were spiked for the AFTIG study, again, different amounts. We see that, again, these are all very similar. Obviously, HPV-18, Tom just mentioned, is in the genome of the HeLa cells. So we really get very similar results across both workflows. So We decided to take this up a notch and perform a larger research study using our live dengue vaccine. These are just non-GMP samples of the dengue-3 recombinant live vaccine, where we performed a spike recovery study using the panel. We also had unspiked samples in order to sort of generate these results and understand the workflows. We're using the short read alignments to a curated clustered RVDB version 25, so late 2022. And then we're following that up with an additional de novo assembly alignment performed in virus screen to compare that to the virus screen short read alignment. And the de novo assembly is performed with MegaHit, and the contigs are aligned reference using Magic Blast. So this is the study research design. Basically what we have is unspiked or spiked at 1 × 10⁵, 4, 5 harvest fluids or control fluids. We have media control, buffer control. We also did a pellet, but I'm not going to talk about that today. So basically here you see how we've extracted it, the kind of library we used, and then the different workflows with Hive or virus screen, subtracting the vaccine, aligning to the database, reviewing the hits, or after we subtract the vaccine, going into de novo assembly, and then going through that pipeline. So what we're able to appreciate from this is that across both virus screen and Hive, we're able to get around the same number of vaccine subtracted reads, and then the same number of reads aligning to the database. So when we look at the spike recovery across the 5 viruses, we can see that either for the control fluids or the virus fluids, we're able to very nicely get hits for 10⁵, 4, and 5 for 4 of the viruses, but not for PCV1. For that, we were only able to detect recovery at 10⁴. We didn't go below 10⁵, so we're just calling that our limit. So again, similar results across both workflows. So looking at de novo, again, just using the control fluids and harvest virus fluids, we're able to detect the 4 viruses pretty well. We have HHV4, HRSV, feline, and reovirus. We're only able to detect it at 10⁴. For PCV1, we're able to detect it at 10⁴ and 5th for the control fluids, but for the virus harvest, we're only able to detect it to the pangolin species, but these are both in the same genus. So this was a kind of a weird result. I wanted to follow that up and see if those other reads from the COVID-2 vaccine we had examined would also hit in de novo, and we were actually able to recover PCV1 there. So it seems that it's got to do with the curation of the database and also probably some sample or read set specific properties. But we were able to detect this, so we were very happy. And so when we really look across the different workflows and pipelines, we're able to appreciate is that Hive is returning more of the species, and we're looking just at the top row here for the virome. If we look at the number of hits for the SRA, it's way greater than the number of hits for the de novo alignments that we would have to go into a counter screen at this point. So if we look at the number of genera, though, when we look at the SRA and the de novo alignments, we have almost the same number. So that's reassuring. So we do think that the Hive is returning more hits because we have a paired read filter that we apply in virus screen and not in Hive. But again, now we're seeing that we do have a higher number of putative hits. So just to kind of go through what a counter screen is for us, basically, so this is a hit that we get to HIV-1. So what does this mean? So right away, I'm looking at a coverage plot, and what I see is that there are 3 places along this reference where there's alignment, and it looks almost exactly the same. So I am automatically pretty much assuming that it's the exact same sequence. I look at another spot of another accession, and it's just stacked up, really short area. So I pretty much already know that this isn't real, but I go ahead and perform an alignment with BLAST-X, and it's really demonstrating best matches to non-viral accessions. So it's just not HIV. Now, what would we do with a real virus? So since we had this pangolin circovirus hit, I figured I would use that as the example. So what we're seeing here is example coverage plots, and it looks like a lot of coverage there, right? That looks pretty real to me. So what do we do from here? So we perform a blast X, blast N. What we're seeing here with the blast X alignment is that we have a best match to the replicase from PCD1. So other alignments demonstrate high matches to several circovirus species. This is the taxonomy tree from BLAST. So, you know, in this case, we didn't get the species that we were looking for, but upon counter screening, we were able to detect it. So, you know, I'll just conclude here and say that, you know, we are undertaking these studies to supplement the risk assessment for live virus vaccines. They can't undergo viral clearance, so it's a little bit challenging. But we've been using Hive bioinformatics for characterization studies filing, and now we have an internal application that we've streamlined this bioinformatic analysis for potential future release GMP materials. We've compared the workflows, benchmarked, generated large data sets to complement the initial comparisons, and then we've additionally tested the end-to-end workflow, which led to the identification of some successes and opportunities to leverage in studies for future filing studies. And then obviously further automation of the largely manual analysis, counter screening and comparative analysis, putting that into the GMP version would be really helpful for the future. So I'll just end with some credits here. Our end users are in Austin, West Point, where I work. Bioinformaticians are in Cambridge, and then our software architects are in Prague. So thank you.
Thank you, Simon. So this is the last talk. It's either the anticlimax or the dessert. Song in my team talked about the haystack, reducing the size of the haystack. But then when the bioinformaticians in my team get it, it's still a haystack. And so I want to talk about how our pipeline has evolved over the years and our general approach to software development. You've seen this kind of slide before. The only point of showing this slide again is to put the bioinformatics in context. It comes at the end of the process, and the entire assay is modular, and so is the bioinformatics. And you're going to see the modules in the bioinformatics. The modules really make it easier to upgrade because there's parts that don't change and there's parts that do. And the input and output interfaces of each module can remain constant so that things upstream or downstream don't have to worry about the impact of the particular module being updated. In 2018, Christophe and others in the AVD-TIG published a considerations paper for optimization of high-throughput sequencing bioinformatic pipelines. And I like to see this, there's too much stuff in there. There's all the possibilities are in there. And so I like to see this as the design space. So nobody's pipeline is going to look like this, but they're going to carve out a path through this design space. If you haven't read this paper, that is a significant achievement from our subgroup D. So PhyloID is what we call our pipeline, and it's an evolving concrete instance within this design space. So we've carved out a path through there. The purpose of it is to identify putative adventitious agents. The focus is on viruses, but it can do more. And the samples that we tend to be in scope for us are cell bank, seed lots, and viral harvests using high throughput sequencing. And specifically, we do Illumina at the time. We're exploring long reads as well. And the development started way back in 2010 as a response to the paper by Eric Delworth's group, where they detected a virus that there was no other way to detect. And so this really brought this technology into focus. We'd been exploring it before, but just sort of, trying it out to see what it can do. And now it was clear that it had a very important viral safety aspect. So 2010, and then the assay, that includes all of the wet lab and the bioinformatics, was finally validated in 2017. So it did take a lot of development work. And back then, people weren't so sure about whether this was the thing to do. And I'm going to talk about that. So this slide looks very strange because there's empty bullet points. And I really could have compacted the flow chart. It's sort of sparse because I'm going to be filling it in as we go along. And so PhyloID version 1 had a Linux command line interface. We recognized that the sequence data were, even back then in the early 2010s, too big. It would be too slow. And so we needed something to compress the data down. And what we chose was de novo assembly. And so this helped to reduce the redundancy. And then the core of the pipeline is our BLAST-N-based screening and counter screening. Back then, there was no RVDB, or it was just, an early version. And so we used RefSeq because it was references that we could trust, and it seemed to be reasonably comprehensive. Now, because RefSeq is not completely comprehensive, our screening and counter screening pipeline needed to be able to look at things that had weak matches, but yet maintaining a good signal to noise ratio. So each one of the 3 iterations I'm going to show you is going to have the 2 slides. And so the core strategy that we published in 2014, as we presented it at the PDA conference in 2013, is based on, so it's called PhyloID because we build phylogenomic distance matrices of all of the reference genomes. We reduce the size, as I mentioned, by de novo assembly. And then the phylogenomic approach to identify the sequences. This essentially looks at the pattern of matches relative to how they should look like on a phylogenetic tree. And so there's some ranked correlation that we use, and this gives us a really good signal to noise ratio. Because in those early days, people were very afraid of opening Pandora's box. We start finding things, everything shuts down. And so we had to be especially careful to not generate too much noise. Sorry. The challenges were to build the reference genome collection because we had to build the reference genome collection and produce these phylogenomic trees. This was excruciatingly slow and laborious. You'll see how when there's a challenge on the bottom of the slide, you'll see it'll be fixed in the next slide. The de novo sequence assembly could theoretically have led to chimeric sequences and misidentification. We didn't really observe that, and we did some studies to show that, but it was a theoretical possibility. The analysis pipeline itself was slow and had to be done one at a time. And the signals needed to be followed up manually because we just did not have the experience to validate that inside the pipeline. Like I said, the challenges below were solved later. And so this was in operation as a validated system from 2017 until 2022. PhyloID version 2.0 was a major upgrade. So you're seeing the flowchart is getting filled in quite a bit. So what we added is in green. So some of the modules were new, the ones in green, and some of them were improved, which is in purple in the left part of the text. So if you ask a molecular biology in my team about Linux command lines, they call that Klingon and they don't speak Klingon. And so we wanted to have a web form on the cloud. The cloud also allowed us to be able to not be constrained one sample at a time. It was much more scalable. We added a quality filter step, as everybody else does, because if you have low quality sequences, they could be misidentified. And then we finally started screening out the host. We didn't do that at first because we were afraid of throwing the baby out with the bathwater. There's viral sequences inside host genomes. And so we wanted to have a system where we could, if we wanted to, mask all of the endogenous viral sequences, filling them all in with N. And so we managed to achieve that. And so now we could do host pre-screening or seed pre-screening. What that meant is we now had a significant reduction in the size of the haystack. There's still hay and there's potentially needles. We also switched to just a regular deduplication without de novo assembly because the tool we were using wasn't really amenable to being installed on the cloud. The screening and counter screening was the same, except the phylogenomic distance matrices now take 2 days on one node instead of 7 months on a cluster. That was achieved with a K-mer-based approach. In the interim, there had been publications about how to build phylogenomic distance matrices using K-mers. We adopted that, and it made an enormous difference. We added some additional modules to put the signals on a tree. We added a confirmation step. So in other words, a lot of the stuff that people were doing manually were now automated, because if you're doing it manually according to an SOP, always the same way, then you can automate that. We then also automated the placing of those signals on genomic maps. Who likes to write reports? So the code, the C++ code writes LaTeX code, which then gets compiled into a report. And then if we need to, there's another pipeline here that's an independent pipeline, which is our nucleotide resolution mapping, or if you like, targeted, the targeted approach. And so this will give you maps and signals, and then this will place them, nucleotide resolution maps from which you could do, PCR primers and so on. So this was a big upgrade. To reiterate the strategy improvements, the cloud implementation allowed parallel processing. Quality filter avoids misclassification. The host filter cleanly subtracts the host, throwing out the bathwater, keeping the baby. The ordinary deduplication just looks for identical sequences and just reduces the number of sequences a little bit more. The K-mer-based phylogenomics, super fast. Automated follow-up of signals, and a semi-automated decision tree. So you're going to see more of that. So the decisions that we had enough experience on to be confident that should be thresholds, et cetera, we put that into the pipeline. And of course, more user-friendly. So faster, more automated. So if I put on my manager hat, I want my team to do more work, but not to do more work. In other words, to get more done, but not do more work. And so that's quite important in these constrained times. The challenges are, it's still a little bit slow. It's certainly faster than before. And it's can work with bacteria, but do we want to spend $20,000 a month to look for bacteria? No. And also, retroviruses, there's other tests for retroviruses, so we're covered there from a viral safety standpoint, but we would like to be better at detecting retroviruses. So that was, again, challenges at the bottom or solutions on the top of the next one. So notice that PhyloID version 2.0 has an end date of 2025. We're in the process of validating the next version, and that's version 2.1. There's one new module. It doesn't look like a big deal. And the previous talk just showed us, the talk before last on K-mers showed us just how powerful they are in terms of speeding up the pipelines. Now, we knew about K-mers, obviously, because we use them for the phylogenomic distance matrices, but I was not happy putting K-mer identification in the pipeline because of the variability in the viruses. They have too many false negatives. So we developed a position-aware K-mer approach. This allows the K-mers to be really short, but the thing is sequences are sequences. So the K-mers are in a certain order. This allows for mismatches, it allows for indels, and it becomes very specific and also very sensitive. So with that approach, you can essentially take the magnet and pull all the needles out of the haystack. You're going to get some hay as well to come along with it. Whereas the previous approach, version 2.0, the host prefiltering, is we get rid of the hay, and then there's still some hay and needles left over. We made some improvements. So on the previous line, I didn't talk about it, but we had introduced RVDB as a secondary screen. But now in version 2.1, it's been promoted to be a primary screen. But I'm also, putting on my manager hat again. RVDB has some things in it that we don't necessarily need for this application. It's there for a very good reason, because RVDB is used for a lot of different purposes. And so here what we did is to use our K-mer approach to mine RVDB for anything that was not well recognized using RefSeq. And so instead of incorporating 1 million RVDB sequences, we really only doubled the size of RefSeq. And then in an iterative process, you add the sequences of the most value from RVDB into the database. You rerun the test. There's still some missing. You rerun it, rerun it, rerun it. I think we do 64 iterations. And then eventually everything is identified that's in RVDB. And so this is good. So RVDB has had enormous value there. This meant that we didn't have to use RVDB and the expensive tBlastX and BlastX in the secondary screen, speeding up the pipeline. Meanwhile, we're continuing to gain more experience, so we have more automated decision trees. We started to, should have done before, started to output information in JSON format because we're contemplating interoperability with other systems later on. And so that is version 2.1 in terms of the details. And then in terms of the strategy improvements, we can now afford to find bacteria. So now bacteria just take a few hours and we get great results. So it's sensitive, specific, and fast. We did not abandon our previous pipeline. It's still the high resolution screening, counter screening, verification. And so that's still employed when you take the magnet and pull the needles and the hay out. Some of those are tentative matches. They get run through the regular pipeline. But now the pipeline is working just with the needles. It doesn't have so much host pre-screening to do. So it's, again, faster. The improved virus database, RefSeq supplemented with RVDB, really helped us to identify more things. And so now retroviruses are being properly detected, whereas they were not before. So it's faster, more affordable, more automated. Bacteria and retroviruses are now in scope. But of course, the story never ends because evolution continues. And so we still have work to do in terms of the background signals, the kitome, if you like. Because I've presented in conferences before, you can improve your sensitivity only down to a certain point, and then you're dipping into the noise. And so to be able to make any more progress in terms of sensitivity, you have to understand the noise. And so we want to work on that. And then there were great talks in this conference about the novel viruses, the unmatched, and the potential approaches, machine learning and so on. And I'm seeing, I think, papers even more recent than the ones that were presented that offer really a lot of hope in terms of identifying viruses that are not well represented in the databases. So to summarize, our first mission was to get the job done, to meet the viral safety commitments so that we wouldn't miss important groups of viruses. We had to mitigate the risks of missing something. And so, because the in vitro tests and the in vivo tests especially, and even the PCR tests would miss things. And this is the problem we were trying to solve. And so we wanted to be extra cautious and have false positives for the subject matter experts to look at. But we also had to mitigate the risks of finding something, in other words, not opening Pandora's box. So you saw 2010 to 2017, it took us a lot of development and design and implementation to get what we were happy with. This involved deep discussions internally and within, and we're so grateful to the AVDTUIWG because those, I mean, everybody is very, it's viral safety is not, I don't think, a proprietary company thing. It's in everybody's best interests. And so we do share not the details, but we share the best practices. And that really is valuable. And the stakeholders needed to be educated. Of course, by doing a good job, we get more demand. And so version 2 was about scalability, because by having Sanofi accept this assay, they keep sending us samples, and it's like, nah, we can't do it. And so there's delays in projects, and so we didn't want that. And so we needed to speed it up, and you saw what we did for that. Also, it's not wise to rely on people with great specialties and expertise. And so if they're doing things in a specific way, then we just automate that. In other words, making the machine the subject matter expert. Of course, people still look at the results in the end, but they don't have to do the routine looking at results. We wanted to leverage the growing experience to improve the decision trees. And then finally, we now are, we continue to recognize gaps and room for improvement, because none of our pipelines are perfect. And so we worked on the gaps, especially in terms of retroviruses and bacteria. Although we're not doing testing necessarily for bacteria, we want to be able to do it. And then, talk to the stakeholders and say, look what we can do. And then they might say, do it. And then, of course, we'll have another problem. And the performance improvements, again, timelines are always tight. And so we don't want to be any kind of bottleneck. And then the future proofing the pipeline. So I have this is a short list of names because there's a lot of other people that worked on this project with us since 2010. But these are some of the most important people. Many of them are still at Sanofi. Some of them were formerly at Sanofi. And of course, the AVD-TWG, especially the subgroup DE, people outside Sanofi. And also, I mean, at conferences, so people that are outside of this set, they also contribute by asking really great questions. Thank you.
Now open the floor up to see if there's questions from the audience here.
Yes, Peru from FDA again. So thanks for all of you to provide such an insightful presentation. I think it gives us a thinking that we really need to start our subgroup C discussion again. Yes. I do have a question for Tom and also one question for Robert. So for Tom, you say that you cannot detect the real by your camera strategy until you do some clarification test. But I'm wondering in the setting of if we don't know the real is there, so how are you going to find the real? Because you find the real by your qualification test, are you purposely finding for the real or you sort of implement some non-target analysis? And in that way, are you seeing your camera strategy as a primary screening, and then you may use this camera strategy to find something that is the candidate, and then you use this candidate to do the BWA or any kind of the aligner in order to speed up your pipeline. And the question for Robert is that I'm interested in knowing my understanding is that you use the RefC as a camera seed to iterate the RVDB, but I'm not quite sure if right now the NC-BAC RefC has all of the viral taxonomies. If not, then probably if you use that as your camera seed, would that be a risk that you will miss the list?
I will explain, but
Is one on? Okay. Yeah, so you're basically right. So indeed, the first step is, analyzing it with Kamer. And you're right, like as I was saying, like not always are, let's say there are like a few reads that belong to Rio 1. And, if it happens to be the case that none of the Kamers are specific for that particular strain, then indeed we sometimes see, as I was saying, like genus or family or something like that. So then the qualification is indeed like a read mapping approach, which is separate from, because we do like modeler development as well. So it's like separate, but there is no pre-knowledge required because we do, we take all the reads that hit the viral database, irrespective of if it was a species result or family, and then we qualify against all of RefSeq, RVDB, and GenBank. So you don't need to know what's in there because you take all your data, irrespective of viral, I guess, where it is in the viral kingdom, and then you map it against that same database that you camerized before. And so then you see some shifts happening from like family to species or species to strain, or sometimes even like, when we take out cell line, there's still some like slip through basically. So we sometimes even see that that a result moves from the viral kingdom to mammalian kingdom. So we see that as well. Yeah.
so sorry. I guess I didn't, is it working? Okay, I guess I didn't explain it very well. So what we do is we super sample the current RVDB to get many, many millions of reads. And then we use our Kamer approach to identify against RefSeq. The sequences that don't match above some threshold, we take the worst matching member of that species from RVDB. We pull it from GenBank, add it to the collection, do this again until we run out. So it should capture all of the taxa that are not represented in RefSeq.
Do we have any more questions from the audience?
For Robert, I'm curious, do you have any off-the-hip metric improvement metrics for ILO-ID through the iterations of it?
Yeah, so I mentioned the 7 months on a cluster to 2 days on a single node for the reference genome collection. This is quite significant because we also had just one cluster when we couldn't run sample analysis while reference genome update was going. So it was inconvenient. And so now it's also offline and it's a simple switch to the new version. So that was huge. In terms of sample analysis, we went from a month to a week. Now recognize that during this time, the databases are growing. And so the performance improvement is better than that, because if we were still using version 1, it would be, maybe a month and a half. Version 2.1, there's a certain point where you can't shave off too much more time because there's some serial steps in the process. It does go a little faster, like maybe 25% faster, but it also costs a lot less because that's, we're a business. And so it's not just about the time, it's also about the money.
Yeah, and what about sensitivity detection?
It's comparable, so we haven't completed our validation, but all of our experiments so far, it's comparable sensitivity, except for things that we now detect that we couldn't, in which case it's much better.
Without any numbers?
Without numbers?
I was just like, what is like, is it comparable to what we've seen? Is it 10 to the fourth in cell background of a million cells, or?
Oh, yeah, no, but by comparable, I mean like a few percent difference in terms of, we might lose a few reads. Because the KMER approach that we're using is protein-based. So intergenic regions, for example, will lose those reads. If they're completely intergenic, if there's no protein at all in them. And so we're losing, a few percent in terms of sensitivity. Of course, version 2.2 is coming, right? So that's where we're going to try to regain any sensitivity that we might have lost.
Just, I might ask a question of Vanessa. So thank you for your presentation. You were able to compare, the existing, Hive approach, platform approach now to the virus screen, like in consideration of the data that you've received now, and comparing the two. Do you have a plan to move forward with one or the other, or have you made that decision yet, or maybe both?
Thank you. Yeah, so I believe, because Hive has so many wonderful tools in it, we'll still continue to probably use it in research space, right? What we're trying to do is move forward with an internal application, really for GMP purposes. So we have to have that, sort of non-GMP in order to perform like these kinds of studies. So, that's where I see this going. And of course, now with Q5A and increasing the kinds of samples and modalities that we'll be working with, then that might also grow as well.
Okay. So you think that your workflow will diversify right into other types of sample types as well beyond what you've evaluated?
Yeah, absolutely. Yes.
So I have a question for Vanessa. So GW Hive, we used to host our RVDB layer probably 4 or 5 years ago. Yeah, they just provide lots of support, and also they are the main initiator along with AFDA regarding of this biocompute object. And I am curious about one thing that might be also beneficial for our other participants here is that since Hive, that is a web-based tool that provide you the web-based interface. So for the biologists who doesn't have the bioinformatics expertise, they can try to use that. But I'm also curious about, because when we are working with GW Hive, I don't know if that is true or not, but we didn't pay anything. So I'm curious about the cost of how much you have to pay for them in order to run the one analysis.
That's classified, Paige.
Okay, that's what I'm thinking about.
So, I mean, I think so. Obviously, anyone is free to obtain an account for Hive, right? That's freely available. But if we wanted to put our own proprietary data, then we have to have security and an agreement with them, which costs money.
Thank you.
Question online.
Yeah, so I have a question from Jasmine, and it's for Tom. How do KMER classification models deal with potential mutations, real or technical, as seen in read sequences?
Yeah, so that's okay. So a couple answers. So the qualification step that we do is using read mapping. So there, you know, we have more flexibility to detect that. But of course, you know, there are always some unclassified reads, unfortunately. So for instance, let's say we take out cell line, that's 99% of your data set. Then, you know, what's left, we screen against the viral database. Some of it might not hit that viral database, right? So then like, what is that? That's usually maybe like a few hundred reads or something. But, you know, we're looking into that. So it could be that it's bacteria because we're not looking for that currently. It could be something from like the UNIVAC database. So we're actually starting to use that as well. So like linkers or primers or something. But yeah, there is always like some unclassified data. But our database, so we currently screen for like 40,000 different viral species and strains. At least we are very certain that those 40,000 are not in the test sample. So yes, there might be something, you know, new there that's not in the database. That's true. So that's something we have to look at. But that goes for our current pipeline as well. That's fully read map based. There is always some dark matter, unfortunately.
Hi, this is Xu Yang from NIFDC. I have a question to ask Robert. You mentioned in the future you plan to do to detect the retrovirus in your plans. As we all know that if we use a human-derived materials, there are a lot of endogenous retroviruses. How to distinguish the endogenous retrovirus with the exogenous? Have you ever do some trials or some designs? Thank you.
Yeah, so what the pipeline can do is to detect the signals, but really what needs to happen is some wet lab work. And so Song earlier talked about benzonase treatment. We can look to see if that impacts. So what we're looking for is actual packaged retroviruses. So let's say that is the goal, because it could be different goals. If that is the goal, then the enrichment should protect them. If the goal is to look for activity within the cell, then also Song talked about stranded RNA-seq, where we can look for expression, look for the signatures. There's still a lot of development work that we would need to do. We're not going to retire our retrovirus-specific assays just yet, for sure. But one precursor to being able to do any of that is for the pipeline to be able to support that. And so in terms of the counter screening step would potentially remove any of these endogenous retroviruses because they're host. So what we do is we give the viruses a handicap, if you like. If it's matching sort of equally well to a retrovirus in RVDB or RefSeq, and it's matching the host, then the virus gets a bit of a boost in its score so that it gets further down the pipeline and eventually gets resolved. And so it's a multi-pronged approach before we can say that we can actually use it for retrovirus safety.
Okay, thank you.
Maybe I can have a quick comments on that. So as Robert mentioned, lots of samples has been now sent to us for sequencing. So we test some for de-risking purpose. We test some human cell lines, and using the upgrade phyllo pipeline, we do see the retroviral signals, but the endogenous one. And then for the experiments we do with and without benzonase, and based on the preliminary result, we can see the signal, the strain signal doesn't change if you compare the sample with and without benzonase. So, and also it's clear that is a remnants of the retroviral sequence integrated in the genome, not an active one. So hopefully we can, further develop the assay and to detect the retroviruses.
I actually have a question, and I don't know if it's to the panel, maybe to the more general audience, or both. Well, we've been seeing the slides where we're showing the different modules and talking about the module in terms of validation. It got me wondering, like, as we're designing these pipeline, how much rework or little to a lot is there when you're thinking about the short versus long reads, or during the transcriptomics and things like that. Are there any thoughts or work that's been, any thoughts from anyone if the modularization really is modularization, or is there a lot more detail to that?
I can say, so, we're in study 5, the FTIC study 5. And so the nice thing about Kamer is that it's read length agnostic. So we did have like other machinery in mind. So thinking beyond Illumina. So yeah, we haven't really done much with it, but we're planning to use this pipeline for study 5.
In terms of our own pipeline, it is adapted for short read, but being modular, not all of the modules will be impacted by long reads. So we would need to look at them and adjust them. We're still exploring long reads in the lab right now.
Yeah, I'll echo that as well. So, I think when it comes to the risk assessment, right, and choosing the kinds of samples and all of that, it'll still remain the same for us, right? So, but then, like Robert mentioned, obviously there'll need to be that part of the sequencing part and all of that will need to be, updated.
Hey, Dave Darmosh, ATCC. Great talks. This is for anyone who wants to talk Kamers. Have you done any work in optimizing the Kamer size in particular? Because obviously with increased length comes specificity at the cost of sensitivity and computational demand.
Yeah, so we tried Kamers of length 4, 5, and 6, and they all performed equally well, surprisingly, but the 6 MERS were much, much faster. And so we settled on 6.
Follow up, have you investigated Kamer frequencies as opposed to just Kamer identities? At which point tetranucleotide frequencies are a surprisingly
Theoretically,
effective
that
mechanism.
doesn't sit well with me. So I don't even, I mean, if we end up publishing this new approach, we'll have to do the head-to-head comparison. That was a non-starter for me. Because the short reads just don't have enough information to do frequency-based Kamer counting. It works well for us. So we do the Kamer counting for the phylogenomics because there we have entire genomes to count with. And so that's enough information to give us good distance matrices.
Thank you.
Yeah, so for us, we just use, because it's nucleotide, so it's different, of course. 35, Kamers has 35, which is the max that we can use. So like, we didn't like test like lower, like 31 or something like that. And we also have not looked at frequency or anything like that.
Okay. So there are no other questions, none online as well. No, look, I don't see Laurent. Laurent's walked out, but shall we end the session a little bit early? And what time shall we come back after the coffee break? I think it's a coffee break now. 3:30? Okay.
Yes.
In half an hour?
Yeah.
Okay,
Yeah.
yeah,
Perfect.
okay.
Mm.
Okay, so
3:15?
3:15, we'll aim to be back here.
Yep.
Ah,
I think to have a coffee break without the coffee is going to be a challenge itself.
oh,
The 3:15
well, we'll give
session.
Laurent the final say.
Come back at 3:15?
It's a little bit too early for the coffee break, but I think in 5 minutes it should be okay. But if you want to continue just discussing 5 minutes, I will tell them that now we are coming in 5 minutes.
We have a question.
So, Christophe, GSK. So question for Tom. So you mentioned 35 for the length of k-mers. How do you handle cases where the reference sequence is not in the database? So you have, if you have a species in your sample, this distant, like 95% identity, which is quite good already, is there a chance to miss it because the length of the k-mer is too long?
Okay. Okay. Oh, is it on?
Yes.
Oh, okay. Yeah, so that's what I was saying earlier, right? So like indeed, we, if something doesn't hit the viral database and, you know, like, and then it would not go into like the qualification step. Like I said, we always have some unclassified data. That's true currently, as well as with this pipeline. But it's like a very low number of reads so far. So like, and I don't really have a number like, oh, if all of a sudden we would have like 0.01% unclassified, that would be an alarm, maybe. I don't know what number that would be. But I do think indeed it's a good idea for us to look into like taking those unclassified reads and move those also to the qualification step and then see if read mapping detects it. But yeah, if it's really not in the database, then I think it's likely we would also not detect it there, because my experience is that like, it's still pretty close given like the diversity that we have in the database. Yeah, it's a valid point.
Actually, I have a list of questions. I just didn't want to stand between coffee and you know, but maybe for Robert, I'm curious on the bacterial identification. Is that thinking of replacing some of the current tests or more as a follow-up? Also, you know, thinking my mycoplasma is a good way of where that would enter the space, because that's a long assay as well.
Yeah, some tests are too long, and also things like mycoplasma can have nucleic acid-based detection, but if they're too specific, you could miss some species of mycoplasma. So high throughput sequencing solves that. Sterility testing, I can't really see it replacing that because it's too straightforward. But mycobacteria, mycoplasma, maybe. And then the sort of an opposite problem than viruses is they tend to be too conserved. So our experience has been we can really only identify confidently at the genus level. However, we can work on that. We're going to work on that to try to get it more specific, because at the protein level, they're quite conserved. But if we get a protein hit, we can always follow it up with nucleotide level matching.
And is there I guess a different challenge in terms of the background, because we hear a lot of bacterial background,
Yes,
even
so,
just
yeah,
in
so we have
water.
explored that. We're fortunately not seeing background of Mycoplasma mycobacterium. There's a lot of Cutibacterium acnes in everything. And it's consistent depending, doesn't matter on the matrix, so it must be coming from the reagents. That's a very common skin bacterium. We see skin bacteria and environmental bacteria sequences, and if we're sensitive enough, we can see their phages. So the background signal problem needs to be solved first before we do any kind of GMP testing on bacteria, for sure.
Cool.
Right. I'm basically asking this question for timing sakes, so if we're good, we're good. So, you know, maybe this is too early to think about something like this, but I'm curious about how your pipelines would handle therapies that are personalized medicine. So if you have backgrounds coming from individual patients as opposed to a well-defined cell line, how does that affect the bioinformatics? Or do you think that it wouldn't? Do you think you would still be fine in terms of that removal step? I just imagine that there's probably be a lot of differences in terms of endogenous things from patient to patient variability. So just throwing that out there.
I mean, we're all very similar, right? First of all. So, I mean, you know, honestly, You know, like I said earlier, we're really using this for a limited number of kind of modalities right now. And so, you know, to think about the expansion of the pipeline for something like that, I think, you know, we would have to obviously adapt. And I think having to undergo a few feasibilities, right, to understand how we tackle it. And I think that that's how we would approach something that's completely different from what we're doing right now, to be honest. You know, if it's just changing a cell substrate or something like that, you know, that in and of itself takes a lot of effort, but it's a conceivable achievement. And I think we'd have to have a completely different mindset and do some feasibility to determine how to attack that, to be honest.
Yeah, and I want to add as well, like when we take out like human background, like for instance, a HeLa cell line or hack or something, we use actually the human genome from the telomere to telomere consortium. So not the actual cell line genome, because we would otherwise take out HPV 18, for instance. And we want to detect that through other means. So yeah, we would do the same thing. I haven't done it, but just use that human genome, I think would work the same. Excellent.